Hello students, welcome to Distinction Learning Platform, and in this video we are talking about urea cycle. And before we begin our lessons, make sure that you subscribe, make sure that also comment in the comment section as well as like the video. In the comment section, you'll find a link that will take you to the website where you find now other courses. Let us begin with urea cycle. Okay? What is this urea cycle that we are talking about? So urea cycle, it is a cycle that is used to make sure that we get excess ammonia or ammonia and convert it into a less toxic substance which we call urea, so that it can be excreted by the kidneys. So let us write what we're talking about. So this urea cycle, we're saying it is a cycle which is very important to get this ammonia, okay, and convert it into a less toxic substance which we call urea, okay. And this urea, remember, is going to be excreted by what we call the kidneys, okay. And the question that you can ask is that why is this process has to happen? Why is this ammonia has to be converted into urea, and the urea should be excreted by the kidneys? Why? Because if ammonia is left accumulated in the blood vessels, it has that ability to go into the brain, and there in the brain it can cause various complications like cerebral edema, comatose, as well as the death of the patient. So what are we simply saying? We're simply saying to say ammonia, which is this one. So let's try to imagine to say if this is the brain, okay, and this is ammonia. So ammonia, if it is left accumulated in the blood vessels there, it has that ability to enter into the brain via certain cells that we call astrocytes, and those cells, they are able to get or convert that ammonia into a certain substance which we call glutamine, and that glutamine, it has that osmotic method whereby it's going to gain now water from the bloodstream, then enter into the what? The brain, causing cerebral edema. So what we're simply saying is that this ammonia can cause cerebral edema in the brain, causing someone to undergo comatose as well as death. All right, so we need to make sure that we remove this ammonia. No wonder urea cycle is very important. Now, the question that you need to ask yourself is, where is this ammonia coming from in the body? Okay, so why is this ammonia coming from? Ammonia will come from different processes in the body, but the most common process that we know is the digestion of proteins. All right, so ammonia mostly comes from the what? The breakdown of proteins. So now let us understand something here. So if we have proteins, if this is the digestive system, now we have proteins here. We know that proteins, they are going to be converted into amino acids. They are going to be converted into amino acids. That's what we know, right? And this amino acid, they can come from the gastrointestinal tract and being absorbed into the bloodstream and taken to different tissues or the organs. So this amino acid, what we're simply saying is that after the digestion of proteins, we're going to have amino acid, and these amino acid, they're going to be taken up by different body organs or tissues. So now those tissues that we're going to talk about, we'll simply talk about the two important tissues that we need to understand. Okay, so the first tissue, this one, we're going to say this is the skeletal muscle. So this is the skeletal muscle. Okay, that is a skeletal muscle. And let's say this one, apart from the skeletal muscle, let's say this is a brain tissue. This is the brain tissue or other tissues or other body tissues. Okay, so apart from the skeletal muscle, now we can have other body tissues, all right, or like the brain tissue. So these important two tissues, they are going to help us to understand how ammonia is going to be produced. All right. So now, since we know that these amino acids, they are going to be taken up by skeletal muscles for various functions as well as the brain tissue. All right. So now let's start with the skeletal muscle. So let's say this is the ammonia. This is the ammonia. So let us use red now. This is the amino acid. So this is the amino acid that has been absorbed by the skeletal muscle. That's an amino acid. Now, this amino acid, for them to be useful, they has to be combined with something that is very important, a certain molecule that you need to be remembering always. All right. And what is that important molecule that we need to remember? This is the name. This one. So amino acid is going to combine with this one, which we call alpha ketoglutarate. It's going to combine with this very important molecule. Always remember that molecule, alpha ketoglutarate. So amino acid and amino acid that's going to be there, because we know that there are a lot of amino acid that the body is going to absorb into the bloodstream and into the skeletal muscles. We've got essential and non-essential. So an amino acid is going to combine with this alpha ketoglutarate and produce something that is very important in the skeletal muscles. Okay, so let us look at the product for this. So this one and that one, they're going to combine. Okay, they're going to combine to produce two important substances or molecules. The first important molecule that's going to be produced is what we call glutamate. Okay, and the other one is, I'm going to abbreviate this one, which is the alpha keto acid. Alpha keto acid. Alpha keto acid. Okay. Alpha keto acid. Again, this amino acid, they are going to combine with the alpha ketoglutarate to produce glutamate and alpha keto acid. Now, this process, it doesn't just happen on its own. There has to be a certain enzyme that's going to make sure that catalyzes this process. Okay, and what is that enzyme? That enzyme is what we call, so we're saying there's a certain enzyme that's going to catalyze this process. Let us use black for that enzyme. We call it aminotransferase. So we call it aminotransferase. Aminotransferase. Now let us understand why is it called aminotransferase. Why is it called like that? Because as you can see, amino, that is the amino group that is coming from this amino acid. So basically what is simply happening here is we're getting an amino group from this amino acid and transfer it to the ketoglutarate so that it can become glutamate and the alpha keto acid. So basically what is happening there is just the transferring of the amino group from the amino acid then to the what? To the alpha ketoglutarate, so that we can have two important products, which is glutamate and alpha keto acid by that enzyme aminotransferase. Okay, so we can have different examples of aminotransferase. So we can have alanine aminotransferase, we can have aspartate aminotransferase according to the type of amino acid that is there. So let's say if we had a good example of that amino acid, like let's say aspartate, we had aspartate combining with alpha ketoglutarate, and the enzyme we're going to use is aspartate amino transfer. So it will depend on the amino acid that is there. Alright? Now, since we have glutamate here, what's going to happen in this skeletal muscle? Now, what's going to happen is that glutamate is going to combine with something which is very important, okay? So, glutamate is going to combine with something here. So, this glutamate is going to combine with pyruvate. So, glutamate is going to combine with pyruvate to produce C, which is something which is important here. It's going to combine with pyruvate. Okay, it will combine with now pyruvate. So let us use black for pyruvate. Pyruvate. So this is happening in the skeletal muscles. Okay. And the question that you can ask is that why is this pyruvate coming from? This pyruvate. So pyruvate is coming from the glycolysis pathway. So if you remember glycolysis pathway, the end product is pyruvate. So pyruvate is going to be produced in the glycolysis pathway that is happening in the muscles. So this pyruvate now, it has that ability now to combine with the glutamate to produce something again which is important. Okay? So if these two combine, we're going to produce now this one, alanine, and this one. I'm going to abbreviate this one now so that we can have much space, and alpha ketoglutarate. So we're going to have now alpha ketoglutarate. We're going to have alpha ketoglutarate. Alpha ketoglutarate. So I'm going to abbreviate as alpha ketoglutarate. As you can see, it's the same molecule that was initially combined with that amino acid. Okay? And this alpha ketoglutarate now, it can go back there and start combining with other amino acids that are now there in the skeletal muscle. Now, what I need you to focus is on this one, on this alanine. Okay, on this alanine. So this alanine, it has that ability to go into the bloodstream there. Okay, then apart from going to the bloodstream, this is the alanine that is here. Okay, alanine. So let us say Al. Okay, it can be transferred from this skeletal muscle, alanine, then enter into what? This organ. This is alanine. Okay, it has that ability now to penetrate from the skeletal muscle membranes, penetrate into the blood vessels. Then it has that ability to go into the liver. So this organ, this is now the liver. So this is the liver. Alright, now why am I troubling you talking about all those things whereby we produce alanine? Because alanine is the major product that carries amino group into the what? To the liver through the bloodstream there up until it reaches the liver. So again, why are we supposed to have alanine? Why are we supposed to have this process to take place? Because this alanine, it has that ability, or it is not toxic in the blood vessels there, because it has what? A certain amino group that is very important. It's the one, it is the carrier form for what? Ammonia. Okay, alanine carries ammonia through the blood vessels and into the liver. Otherwise, remember what I said is that if ammonia is not carried through alanine, because if it is in the blood vessels there, it can be taken to the brain, causing cerebral edema. So we don't need ammonia in the blood vessels, but we need ammonia in form of alanine so that it can be now transferred to the liver safely. So now, what do we call this now? We call it alanine nitrogen transport. Okay, so what do we call this? How skeletal muscles transfers the ammonia to the liver is through alanine. And what do we call this transfer? transport, we call it alanine nitrogen transport. Okay, so we go to the brain tissues. So on the brain tissues, it's more like the same thing. Now we're going to use another transport for the brain tissues. All right. Okay, now since we know how the skeletal muscles transfer ammonia to the liver via alanine nitrogen transport, now we can understand, apart from the skeletal muscles, how do the brain tissues or the body tissues transfers also ammonia in form of a certain what? In form of a certain transporter. What is that? In case you are examined during an exam. So how are we going to do that mechanism? It's just the same thing. So we're going to say the first important thing that we talked about is this one. It was this one. We said we're going to use red. We said going to write these amino acid, they went into the skeletal muscle, but they can also go into the brain tissues. So these amino acid from the digestion of proteins, they are there in the brain tissues, okay, or in other body tissues. So these amino acids now they can combine with what? We know what is the initial thing that can combine with the amino acid. We said alpha ketoglutarate. That's what we said, right? So that's the recap of whatever we're talking about. If this happens now, what do you think the product will be? What do you think the product will be? Let us use black. So we said now we're going to have glutamate. We're going to have glutamate. And what? We're going to have the acid, alpha keto acid. So alpha keto acid. Okay, let's just say alpha keto acid, Ka. All right. Now the most important thing is the glutamate. So we're going to start using now the glutamate. Initially here, if you look at here, we used glutamate so that it combined with pyruvate to have alanine, so that alanine can be transported via the blood and up to the liver. So the same applies here. Now, here what's going to happen is this glutamate is going to combine with something else, because this is not now the skeletal muscle, it is the body tissues, now other peripheral body tissues. So it's going to combine with now ammonia, free ammonia. So we know that, okay, we know that the body tissues, they are going to be generating, if they are working, they are catabolizing amino acid or excess amino acid. Basically, they are going to have a certain amount of ammonia inside there. So if there is ammonia inside there, that ammonia will be combined with the glutamate. Okay, will be combined with glutamate to produce something which is important. Okay, what is that something that's going to be produced? Glutamine. So we're going to use blue for this one. Glutamine. Glutamine. So we're going to have what we call glutamine. All right, glutamine. Now, glutamine has that ability now to be transported into the blood like alanine. So this is glutamine. This is glutamine that is coming like that. And it has that ability now to penetrate the liver like alanine. Glutamine. Okay, that's glutamine. It's like acting like alanine, right? So, and it is less toxic. So the reason why glutamine, it is within the blood vessels, it is less toxic compared to what the free ammonia that we talked about. Because ammonia, if it is in the blood vessels, it can go to the brain and cause cerebral edema. But glutamine can't do that. It is safe and can be penetrate different membranes of body tissues. So it has that ability to also penetrate the liver and so that it can continue to the urea cycle. All right. So now we've understood how the body tissues or the skeletal muscle as well as the other body tissues transfer ammonia up to the liver safely, because ammonia shouldn't be alone in the blood vessels because it causes various complications. Okay, so now since we've understood about this, Now let us understand something about this part now. So why is glutamine going now? So glutamine, it has this ability to enter into these mitochondria. So this part is what we call the mitochondria. Mitochondria. So this is the mitochondria in the liver. So this is the liver cell, which is the hepatocyte. All right. This hepatocyte, it has two parts. It has this mitochondria, which is important or crucial in the urea cycle, and it has this cytoplasm. Okay. Because if you look at the urea cycle, it's going to have two important parts. There are some processes that will happen in the cytoplasm, and there are some processes that will happen in the mitochondria. All right. So now, what we're simply saying here is glutamine, it has that ability now to go into the mitochondria. Like that will come to alanine. What's going to happen to alanine as well? So glutamine, it has that ability to go into the mitochondria there. Then what's going to happen here to glutamine is that it's going to be converted into something that we need. So glutamine is going to be split. Okay, it's going to be split into something, which is it's going to produce ammonia, okay, which we need, plus now the glutamate. So let's put glutamate to this side. So I'm going to say ammonia plus glutamate. Okay, let us write well. Glutamate. Okay, so what we're simply saying is that this glutamine, the moment it enters into the mitochondria of the hepatocyte or the liver cells, will be converted into glutamate and ammonium. Ammonia or ammonium is just the same thing. Okay. Now, what is the enzyme that is responsible for converting glutamine into glutamate and the ammonium? And this ammonium is something that we need to convert it into urea. All right. So, and this is what we call glutaminase. Okay. The enzyme that's going to catalyze this reaction, we call it what? We call it glutaminase. So we call it glutaminase. Glutaminase. All right. The enzyme that's going to catalyze this reaction where converting glutamine into glutamate and the ammonia, we call it glutaminase. All right. Now, for alanine also can be taken into the what? It can be taken into the mitochondria, and there will be conversion of alanine into something. So what's going to happen is that this alanine as well, it has that ability now to be taken into the liver, and it's going to be converted into what? It's going to be converted into glutamate, as well as another important factor, which is the pyruvate. So pyruvate, I'm just going to use P. So alanine, it will be taken into the mitochondria, then it's going to be converted into what? Two important substances, which we are calling what? Glutamate as well as the pyruvate. Okay, that's the important part. Now, the important thing that you need to understand is that this glutamate now, it also has something inside, which is the ammonia molecule that we require. So we need to break down this glutamate as well, so that we can have ammonia, more ammonia, so that those ammonia molecules can be converted into urea, the urea that we want. Okay. And before we progress, we need to know the enzyme that's going to convert alanine into glutamate as well as the pyruvate. What is that enzyme? We call it alanine. So what do we call that enzyme? So we're going to use red. We call the one that's going to make sure that split alanine. We call it alanine. Let's just say use a alanine amino transaminase. transaminase. Alanine amino transaminase. So that's the enzyme that's going to split alanine into pyruvate and the glutamate. Okay. Now, since we've started the urea cycle now, we need to understand something now. Because urea cycle has to start with now ammonia. Alright? So now before you go to this urea cycle, we need to understand to say urea cycle is going to have two important parts. The processes that will happen in the mitochondria and the processes that will happen in the cytoplasm. So now let us write something about that. So we are saying this urea cycle now is going to have two important processes. So processes that will happen in the mitochondria, okay, and processes that will happen in the cytosol or cytoplasm. So let's just say cytosol. Now, how many processes that are going to happen in the mitochondria? There are about two. How many processes that are going to happen in the cytosol? They are going to be about three. So what we're saying here is that in the mitochondria, we just need two processes to happen in the mitochondria. Then we just need about three processes that will happen in the what? Cytosol. So the total number of processes for urea cycle has to be about 5, which is 2 plus 3. It is about what? 5 processes. Alright. So now since we've understood about that, now let us know how glutamate is going to produce what? Ammonia, because the only thing that we need is this ammonia. Okay, this ammonia is the one that we need here in the mitochondria. Now the question that we can ask is that how do glutamate produces that ammonia? So glutamate is going to be broken down, okay? It's going to be broken down by a certain enzyme that we write here, okay? So this glutamate is going to be broken down into ammonia or ammonium ion with a certain molecule that's going to be produced, which is alpha ketoglutarate. So we're going to have alpha ketoglutarate here. If the moment glutamate is being broken down by a certain enzyme that we talk about, then we're going to have alpha ketoglutarate and the ammonia that we need, right? And what we call that enzyme, so make sure that you be taking note of these enzymes. So the enzyme that's going to catalyze this reaction, what we call it? We call it glutamate dehydrogenase. So we call it glutamate dehydrogenase. Glutamate dehydrogenase. Dehydrogenase. Okay, this is the enzyme that's going to do that. All right, glutamate dehydrogenase, the enzyme that's going to break down this glutamate into alpha ketoglutarate and the ammonium ion that we need. Okay, now since we've understood about that, now here that's where urea cycle is going to start now. Here on the ammonium ion there. Okay, that's where the urea cycle is going to start from now. Okay, so what's going to happen here is this ammonia will start combining with something. So before you go to the urea cycle details, then we need to write the molecules that we need to be following. Okay, so let us list the important molecules that are going to be what produced there, so that we can be following step by step. So we're going to list the first one. The first molecule, we know that it is ammonia, which is there. Okay. The second molecule is what? The second molecule, we know now it is I'm going to write in, I'm going to abbreviate this one. We call it carbomoyl okay, phosphate. Okay, it's just fine. So carbomoyl phosphate, that is the second molecule. So make sure that you take note of that. Carbomoyl phosphate. The other molecule that's going to be produced is citrulline. So we'll be following this. Citrulline. Okay, this is the other molecule that's going to be produced. Number 4, another molecule that's going to be produced apart from citrullin is arginosuccinate. Arginosuccinate. Succinate. Okay, arginosuccinate. Okay, arginosuccinate. Then the last one is number 5, arginine. Arginine. Okay, so remember what we said is that how many processes that are going to be involved in the urea cycle? There are about 5. There is the ammonia, the first step, carbon monophosphate production, citrullin production, arginosuccinate, and the arginine production. So now let us start with the first 2 processes that happen in the mitochondria. Okay, the first 2 processes that happen in the mitochondria are this. This ammonium that has been produced will combine with something, will combine with bicarbonate, okay, will combine the bicarbonate and with ATP, okay. So this is the first important step that's going to happen. So I'm saying this ammonium that has been produced because of these processes that we're talking about, they will start combining with bicarbonate with ATP, okay, and to produce a certain important molecule, which is what? Carbomoyl phosphate. So I'm going to say carbomoyl phosphate. I'm going to abbreviate, which is the CP, carbomoyl phosphate. So again, ATP, bicarbonate, and ammonium, they are going to combine together to produce what we call carbomoyl phosphate. And the enzyme is this one. The enzyme that's going to catalyze this process is what we call carbomoyl phosphate synthetase 1. Okay? CPS1. Carbomoyl phosphate synthetase 1. This is the enzyme that's going to catalyze this reaction in the presence of ATP. And how many ATP that are there? There has to be 2 ATP that are going to be used up here. So 2 ATP are going to be used so that we can have carbomoyl phosphate. Okay, and the enzyme that's going to catalyze this reaction is carbomoyl phosphate synthetase 1. Okay, and this process, the first process where you are producing carbomoyl phosphate, is what we call the limiting step. So this is the limiting process, okay? One is a crucial process. Minus this process, the urea cycle can't happen. So the other important concept that we need to understand is that this process is the what? Limiting step. Which simply means that minus this step, the urea cycle can't happen. Okay, it's the one that determines if the urea cycle will start or is going to stop. Now, this CP is going to be converted into something within the mitochondria. Okay, what did we say? It's going to be converted into citrullin. Okay, it's going to be converted into citrullin. Citrullin, but there has to be this CP, which is carbomoyl phosphate, has to combine with something. Okay, this one has to combine with something because it doesn't just going to be converted into citrullin on itself. It has to combine with something. What is that? We call it ornithine. We call this molecule as ornithine. Ornithine. Okay, so ornithine is going to combine with the carbomoyl phosphate to produce citrullin. Again, this is going to require a certain enzyme. So make sure that you take note of the enzymes. The first enzyme that we've talked about, the most important one is CPS1, which is carbomoyl phosphate synthetase 1. Then the second one is this one that will happen within the mitochondria. What is that enzyme? We call it now ornithine. So we start with this one, ornithine. So I'm going to use red for this one. Ornithine, carbomoyl. Ornithine transcarbomoyl. So we use T, transcarbomoyl. Amylase. Okay, ornithine transcarbomoyl amylase, that is OTCA. Sometimes they just say OTC. Ornithine transcarbomoyl amylase. Okay, that is the enzyme that's going to catalyze this process, whereby you are going to combine ornithine plus carbomoyl phosphate into citrulline. Now, this process basically it will make sure that there is production of citrulline, and citrulline, it has that ability to come out of the mitochondria or to pass through the membrane, cell membrane of the mitochondria and goes into the cytosol. So what we simply mean is this citrulline can go into the cytosol here. It can go into the cytosol. This is the citrulline. Why is it doing that? Because it can penetrate the cell membrane of the mitochondria. So that's why it is able to come out of the mitochondria and go into the cytosol or the cytoplasm of the liver cells. Okay, now what's going to happen here? Citrulline will start combining with another molecule. So what is that molecule? Citrulline is going to combine with what we call aspartate. It's going to combine with what? Aspartate to produce something. Okay, to produce this one. Aspartate to produce what we call this one, which is the agenosuccinate. Agenosuccinate. But this step is going to require ATP. So it's going to require what we call ATP. It's going to require 1 ATP. It's going to require 1 ATP. So remember that here we had about what? How many ATP? 2 ATP. But here we are only using up 1 ATP. Okay, to produce agenosuccinate. But again, remember that each step requires a certain enzyme. Okay, and this enzyme is what we call agenosuccinate synthetase. So just get this word, agenosuccinate. So agenosuccinate, you're just going to say this is agenosuccinate, then synthetase. Okay, agenosuccinate synthetase is the one that will use 1 ATP and combine aspartate with citrulline to produce agenosuccinate. Alright, again we proceed. Agenosuccinate is going to be converted into another important molecule. If you remember, if you follow this one, we're going to have what? Arginine. Okay, we're going to have what? Arginine. So let us use black. So agenosuccinate will be converted into arginine. Okay, arginine plus a certain molecule that is also important, which we call fumarate. So you need to understand here that we're going to have 2 important molecules that are going to be produced, which is going to have fumarate plus arginine. Okay, so if we have fumarate and arginine, now we're going to make sure that arginine is going to be converted into something as well. Okay, so now what is the enzyme that's going to convert agenosuccinate into arginine? What do we call that? Remember what I said is that any step is going to require what we call an enzyme. Okay, so this one is going to require what we call arginosuccinate lyase. So the enzyme that's going to catalyze this reaction is what we call arginosuccinate lyase. Arginosuccinate lyase. Okay. Lyase, or sometimes you can just say arginosuccinase. So arginosuccinase or arginosuccinate lyase, that's the same enzyme. It's the one that's going to break down this arginosuccinate into arginine. All right. Then what's going to happen is that after this, so this arginine will be converted into urea and ornithine plus ornithine. Okay, plus ornithine. As you can see, urea now, it can go to what we call blood vessels. So it can go into the blood vessel so that it can be excreted by the kidneys. But this urea now, since it can be excreted by the kidneys, now we've got another important substance or molecule, which is ornithine. Ornithine can go back into the mitochondria and combine with what? This molecule, which we said carbomoyl phosphate, so that we can have now citrulline, so that the cycle can continue like that. Okay, so ornithine, it has that ability to go back. Okay, and the enzyme that's going to catalyze this and the breakdown of arginine into urea and ornithine is what we call arginase. Okay, and the important part is here there is a certain important molecule that is going to be produced after arginosuccinate is going to be converted into arginine by the use of arginosuccinase lyase, which is fumarate. Fumarate is an important molecule that's going to participate in the Krebs cycle. Okay, now, or TCA cycle. Now, since we've understood to say, now the byproduct that we have is urea. Urea can be taken into blood and excreted into the kidneys, but the only thing that's going to be produced here will be now, will go back into the mitochondria and combine with the CP to produce now citrulline, so that the cycle can go back again. All right, so that's where now the urea cycle ends from. Okay, but there are some various disorders that you need to understand. So let's talk about the disorders that you need to understand. So there are some various disorders that you need to understand. And you're going to be examined on this. So disorders that you need to understand is that we can have enzyme deficiencies. Okay, we can have enzyme deficiencies. So the first important enzyme deficiency is this one, CPS1. Okay, CPS1, who can remember that? Can you remember that one? CPS, that is carbomoyl phosphate synthetase type 1. Okay, carbomoyl phosphate synthetase type 1. But there are some disorders where you are going to have deficiency of these CPS1 enzymes. Okay, and if you have deficiency of CPS1, basically you are going to have what? The buildup of ammonia in the blood vessel, causing cerebral edema, comatose, and death. So the first disorder is the deficiency of what? This enzyme. Okay, the deficiency of this enzyme, and we're saying this can lead to what we call cerebral edema, confusion, coma, death. Okay, which is very, very dangerous, okay, or fatal to someone. Then we can have also another important deficiency, which is the deficiency of that enzyme, which is ornithine transcarbamylase enzyme deficiency. So we can have also ornithine transcarbamylase enzyme deficiency. Okay, we can also have this deficiency, and it can also lead to what? The same complication, which are these complications that we're from talking about. So the goal, or the thing that we need to understand here is we can have two important enzyme deficiencies which is what? This one, the carbomoyl phosphate synthetase type 1 deficiency and ornithine transcarbomoyl phosphate deficiency. Okay, this one amylase, I mean ornithine transcarbomoyl amylase deficiency. This one they can lead to cerebral edema, confusion, coma, death as well. All right, but the only difference between the two is that this one, it is an X-linked disorder. This one, the only difference that I can put here is it is an X-linked disorder. Basically, it's common in what? Females. This one. Common in what? Females. So these are the things that we just have to remember. And this is where we end from. Okay? And make sure that you comment, like the video. All the best until next time.